So our next speaker is Adam Ferguson. He's a professor of neurological surgery at UCSF, and he's also a PI at the VA, and he leads a brain and spinal injury center, BASIC. So Adam is involved in UCSF REACH. He's a co-investigator, and he provides his expertise across several research cores of UCSF REACH. And the reason being because his expertise is extensive. So he has two different lives. One is the basic animal research in pain, where he focuses on traumatic brain injury and spinal cord injury. And his second life is bioinformatics, where he runs, where he actually pioneered the collection of big data to understand veterans' health, especially as it relates to spinal cord injury and TBI. So today we're going to actually, we're going to be introduced to the work he's doing using multimodal analysis to understand precision medicine. So I welcome Adam to present.
Thank you. Thank you guys for having me. Can you hear me well? Yeah, okay, good. So I have some disclosures. Most of them are federal grants and a few kind of nonprofit and in-kind support things that I won't be discussing in detail. So my outline for today is to describe biomedicine as a big data problem. I think this is a theme we've touched on quite a bit this morning. I'm going to then provide a few examples of machine learning and artificial intelligence in action in the context of biomedicine, specifically around neurotrauma, which is kind of my natural home. And I'll close with an example of automated machine learning for decision support and how that's actually guiding rapid translation. So the premise of the talk is that orthopedic pain is complex. Which I think may come as no surprise to the folks on this group. The idea here is that modern disease classification is a mixture of anatomically, physiologically, metabolically, immunologically, and genetically defined factors. And together they describe the individual that's afflicted. And in the preclinical literature, the basic science literature, this manifests as gazillions of tiny measures of biofunction. So the literature is filled with these multi-panel figures where you show changes in multiple organ systems and then how those manifest functionally. And what I'm going to be arguing today is that we need to move beyond these multi-panel figures and as a field, try to bring our data together into a common data ecosystem so that we can operate on it with machine learning and AI tools. And this sort of recasts injury, not so much as a biomedical problem or a biological problem, but really as a problem of big data. And this term big data, those of us in the Bay Area know, is on billboards everywhere. So I just wanted to pause for a second and describe what that buzzword actually means. It comes from Gartner, which is an IT firm, in the early 2000s, where they described the big problems of modern data management as being comprised of the 3 V's of big data. This is the problem of large data volume, of high data velocity. So this could be small data bits that are moving very quickly, like the Twitterverse or the modern ICU. And then the problem of wide data variety. And I think if you think through pain, generally, it really is a problem of wide data variety. You know, and in some ways, it's a microcosm of a broader problem in biomedicine of our need to datafy biomedicine. So if you look across the published literature, you see that there's a great many data sets, and actually a relatively small number of them are comprised of organized big data. We have things like the Human Genome Project, maybe some of the large imaging repositories, but most of the data that actually appears in the literature is modestly sized and falls out in this long tail of the data distribution. So these are a great many data sets, not all that big, wildly heterogeneous, high variety, and hard to manage from a data perspective. At a certain point, you reach the literature limit, and beyond that, you have this unpublished and dark data tail of the data distribution. We now know from meta-analyses that this comprises about 85% of the data that's collected in biomedical research. So that has some odd economics associated with it because worldwide, we perform about $240 billion a year in biomedical research. These are 2014 dollars. And of course, this means that every year, about $200 billion of our biomedical research investment is collected and then placed in a file drawer and not disseminated in any form, which is somewhat wasteful, you could say. And this can really be attributed to the way that we do data sharing in biomedicine, which is this 17th century data sharing technology, the scientific paper. It's a narrative-based way of disseminating knowledge. And so there's been a big push to develop more modern versions of data dissemination in biomedicine. And this has been encapsulated in the fair data stewardship principles that have been championed by the NIH and are a major portion of their 2023 data sharing policy. The idea here is that data themselves should be made findable, accessible, interoperable, and reusable. So rather than actually collecting data and then needing to write about it, we can collect data and hopefully the data sets themselves will become academic work products that are disseminated. And people will hopefully be promoted and get tenure based on the quality of their data rather than the quality of their storytelling. The spinal cord injury field has actually been an early adopter of these fair data principles. As a community, because it's very heterogeneous and spinal cord damage is not solved, there's no treatment for it. We've gotten together and sort of sketched up what the issues would be for us to make data fair. And this has culminated in a open data commons for spinal cord injury that's a publicly available repository. And based on our polling in the community, we found that people are very willing to share their data so long as PIs have control over the data, according to their legal responsibilities at universities. So we have built this ecosystem where people can deposit data in a private lab, but then very easily the PI can right-click and decide how the data are shared. They can share to labs in a community space, and ultimately through the California Digital Libraries, they can provision a digital object identifier and then have their data released on a CC BY attribution license, which then provides citations for data. So if someone reuses their data, they get credit, they get their citations for those data. So I want to close the rest of my talk with a few examples of what you can do once you have this type of big data variety in one place. So the basic principle here is that we should be able to apply machine learning based data integration to complex data. And I think this picks up on the themes that Keith mentioned at the start of the day. So the idea here is that function after low back pain or orthopedic trauma is not one thing. It is really multifaceted, and you can conceptualize that here in a two variable space by looking at the reported disability, which might be a clinical measure, and then physiology. And ultimately, you can place an individual in their unique position in this two variable space or bivariate space. This individual can be a human or a model organism. And ultimately, your question is, how does that individual stack up against the population of all other individuals? And you can, of course, do this across multiple layers of knowledge. And if you track the same individual across functional changes, biomarkers, and tissue changes, you can then draw these linkages that allow you to very rapidly cluster individuals based on their similarity in a multidimensional way, and then understand what it is to be a member of a particular patient group. And then, of course, this becomes a tool for precision medicine because you can ask, to what extent do these individuals overlap or are they separable in a multidimensional outcome space? So that same concept can be applied in translation and reproducibility testing, and then, of course, in rapid precision medicine contexts. And the goal here ultimately is to come up with a predictable response where with an N of 1, you can determine whether an individual patient is going to respond to a drug or not. So I want to give a couple of concrete examples of this to close out my talk. And these come from these UCSF initiatives, the Transforming Research and Clinical Knowledge in TBI study, which is a very large observational clinical precision medicine study, and then Track SCI, which is a smaller effort at the Zuckerberg San Francisco General Hospital. So I'm going to start with Track SCI. There's a project that was undertaken by Jason Talbot, who's a young neuroradiologist, and Jenny Hafley, who is a clinical data scientist and assistant professor now at UCSF. And they were concerned with the problem of MRI imaging of spinal cord injury. And ultimately, Jason pointed out that there are many different ways to score MRI lesions after spinal cord injury. There are many types of abnormality. It's not very clear which of these is the most true representation of the injury. So rather than make a strong claim about that, he and Jenny developed this machine learning approach using nonlinear principal component analysis, where they pulled in all the data, did optimal scaling transformation determined by a machine learning tool, and then pattern detection. And out the other end comes this linear principal component score that they could then use to predict outcome. And what they saw is that the MRI space stratified into these two separate clusters. And then if they projected individual patients from the Zuckerberg San Francisco General Hospital into this two variable MRI space, they could very quickly determine how these these humans were going to land at discharge with respect to their impairment. So this was kind of a remarkable finding because they had MRI in the first 4 hours, and they could predict where these patients would end up in their impairment down the road. A very similar type of narrative can be told in the Track TBI project. Esther Yu, a radiologist, and Jeff Manley, a neurosurgeon, developed a way to predict long-term outcome in TBI. Long-term is 3 months after injury, just by looking at the acute MRI signals. And this led to the first ever FDA-approved biomarker for traumatic brain injury, precision medicine. So the idea here is that you can actually use this very specific MRI sequence early on and then project where patients are going to land and actually stratify patients based on this early imaging. So the problem with this idea of using machine learning and AI for biomedicine really, I think, comes down to this issue that if, say, Connor wanted to apply these tools that I've described, we say, that's wonderful, Connor, but you're going to have to take a bunch of stats and computer science and, coursework and actually transition from being a clinician to a data scientist. And pragmatically, that's going to be hard to scale up. So I summarize that idea as advanced analytics are not really for hobbyists. So to solve this problem, we need to come up with tools that make machine learning more automatic, more user-friendly. And so this is where partnering with advanced analytics entities, companies, can help. So as one example of this, we formed a partnership a couple of years ago with a group that spun out of Stanford's math department, where we're able to take in multi-species data, and we can render it in a computational cloud into a correlation space automatically, and then apply a mathematical lens automatically that will project individuals into the multidimensional syndrome space described by the disease. In this case, it's spinal cord injury. And then we can sample this space many times and come up with an ensemble cluster algorithm that shows individuals that are similar as nodes, and clusters that share at least one patient in common are represented by edges. And then what emerges from this is this massive topology that represents the multidimensional space of the disease. And so that sounds like a lot of stuff, but it's really a click button. You go boom, and then this map unfolds of the topology. And then you can rapidly explore the map by painting it by variables of interest. So just to make this more concrete, I want to show some results from a paper where we deployed this tool in spinal cord injury and traumatic brain injury. And the example I'm going to use is really ancient multi-center animal spinal cord injury data that was in paper form. So we ingested all these data from a multi-center preclinical animal drug study. And very quickly, once we had the data in a digital form, we could say, how does locomotor recovery manifest across all of the variables measured? And we saw that there are some animals who did very poorly and some who did well. And then we could very quickly say, okay, what are the features that seem to be driving these group differences? There are thousands of variables that we fed this. And it turns out that the single most important variable for predicting long-term locomotor outcome in this context was the blood pressure of these animals on the table during the surgery. This was a very wild finding that the original authors were unaware of. And basically what it was is that animals with very high blood pressure had very poor long-term neurological recovery, which is a wild finding that apparently our neurosurgical colleagues weren't aware that this was a really big issue. So this has led to some clinical studies where we're going through the question of whether blood pressure predicts outcome. And it actually has led to some reproducibility studies where we've gone back and tried to scrape all the remaining data that we could find from these animal studies. And we now have generated a paper where we have released 1,400 animals from this original study and made the data fair from the 1990s to drive more machine learning discoveries. So that was a kind of wild ride, but I was hoping to demonstrate ways that we're applying machine learning to both drive new clinical discoveries and also ways that we're using sort of modern data science to make dark data available for reuse. So I'll stop there and plug the places I work and the many people I work with.